in order from coffee, from tobacco, potato, or something like this. We want to find some alternative for this intercalating agent against virus. Firstly, we want to test the alkaloids into the Swiss Army to see the Lipinski rule of 5 property, if they obey it or not, and then to check the bioavailability if the metabolite is able to reach the bloodstream immediately, and synthetic accessibility, which refer to its complexity and if it can be built in a synthetic condition in an easy way or hard way, which means related to in order to synthesize this molecule. For the molecular docking, we use Pyrex autodocvina. For the RNA receptor molecule, we process it using open Babel because the built-in open Babel in Pyrex could not recognize and process the RNA molecule because they are I think they are only designated for the protein molecule receptor only. For the metabolite ligand, we use the built-in open Babel in Pyrex to minimize the energy using the UFF setting. We then determine the docking space before we conduct the docking. Follow up the docking, we validate the docking scoring using Annapurna, visualize the molecular docking using complex using the PyMol, and for the bond analyzer identification, we use PyMol, Discovery Studio, and Finger RNA-T, which is currently in preparation because it's not been developed completely by Bochnicki lab. And for the result, of all the metabolites, solanine and tomatin do not follow the lipid rule of 5 because their molecular weight and the hydrogen atom donor and acceptor. We have to use some some way for drug delivery, especially their bioavailability score is very low, in order to reach the viral infected target. The synthetic accessibility is very large, as you can see in tomatin is 10. Is the end spectrum, which means that tomatin is really hard to synthesize, which we need to talk about that later. And solanine and tomatin has also the most negative docking score here compared to berberin, which means tomatin here especially could serve as a potential intercalating agent for the viral RNA. Consistent with the docking score, we see using finger RNA-T, the solanine and tomatin, especially tomatin, has a lot of hydrogen bonds. However, the other bonds has not been identified using finger RNA-T, and we need to understand why for this, and we haven't found it for now. This is the bond visualization. The number 1 is for 3-dimensional, and number 2 is 2-dimensional using Discovery Studio, and 3-dimensional one using PyMol. To discuss about it, so yeah, tomatin has the most negative, the strongest bond to the RNA, followed by solanine. As they're using the finger RNA-T screening, the hydrogen bond is also abundant. So far, finger RNA-T reveals more hydrogen bond, which is consistent to the docking score, indicating that the reason behind why the respective molecule is strong enough to bond to the RNA. But the other bonds, except for lipophilic bond, which have been identified by Discovery Studio, has not been revealed by finger RNA-T, and this requires validation. The drawback for tomatin and solanine, they do not follow the lipid rule of 5 because with the trial violation. They also have low bioavailability and very high synthetic accessibility. They need some special way for drug delivery method to reach the viral infected target upon the infected tissue area caused by the virus. We need to optimize the way for RNA ligand docking in the future to improve, to see how the RNA and the ligand molecule interacted with each other. We need to identify how the bond is between these two molecules. We need to optimize the drug delivery specifically for viral RNA target. And also we need to test it in the in vitro experimentation in the laboratory. So in conclusion, the best candidate so far is tomatin. However, we need to formulate the way for drug delivery to reach the viral infected area. and also the way to synthesize the molecule if we want to do it synthetically. And also, based on our study, Pyrex can be used for docking, supplemented with Open Babel for RNA receptor preparation. And for visualization, we use PyMol and Discovery Studio. But PyMol, I think, is the best for because it's 3D view. And finger RNA so far can reveal most hydrogen bond, and so far it's promising for us. So this is from us right now. Thank you. Please don't hesitate to contact us in the future if you want for future collaboration and any other study. Thank you very much. Terima kasih.
Thank you very much, Aditya. Thank you for the presentation. So let's continue to the question session then. Professor Yelikci, do you have any questions?
No, thank you very much. It was a very clear and nice presentation. I congratulate the person. So they did a very nice job.
Okay, thank you very much, Professor Yelikci. Dr. Serçinoğlu, are you there?
Yes, thank you
Do
for
you have
this
any questions?
Yes, I have just one. So you have tested something called synthetic accessibility for these compounds. So what does that mean? Can you explain?
Well, from the literature that I read, synthetic accessibility refers to the complexity of the molecule, which means if the synthetic accessibility scoring is really low, the molecule can be synthesized outside the, well, basically can be synthesized rather easily. But if the synthetic accessibility in the end gradient, I mean in scale of 10, which means the molecule is very complex and it requires a long way to biosynthesize the molecule. That's what I learned, and it will refer on if we want to make it synthetically outside the living condition in plants itself. That's what I learned.
Okay, then why would you need to synthesize a natural compound then? Just purify it from its natural resource. So well, it seems this is more relevant for synthetic small molecules that are designed from scratch in lab and not for natural products. So on that account, I found it a bit confusing. Why you have ever considered synthetic accessibility for these compounds? I found it a little unnecessary. That's my opinion.
I think I agree with your opinion. Thank you for your input. I will reconsider for it in the future. I will perhaps, yeah. I think I just want to view if this in the whole plant, especially for the tomatin that we've used for this screening, can we just get this metabolite rather easily because it's can be made in an easier way in the biosynthesis, or it will be a long way. Okay, but thank you again for your input.
All right, fair. Thank you.
Yeah.
Okay, thanks. Then I have some questions. Dr. Serçinoğlu, do you have any more questions?
No, I don't have any.
Okay, then I would like to ask some questions. If I understand correctly, you want to target RNA against COVID-19, yeah? So do you think that's a good target?
Well,
I was a little bit surprised, but do you think it's a good target?
Honestly, I haven't considered it that much, but I just wonder if there are any other kind of possible cure or not cure, basically, any possible drug that we can use against COVID, because we know that there's other targeting, because I mean we normally use protein for this, but just in case if the metabolite can be good enough against the RNA molecule, which can be used for any other potential use in the future, it can perhaps it will be a good idea. That's what my opinion is.
Okay, so you think RNA is a good target?
Probably.
Okay, so what do you think about the lack of stability of RNA molecules then? Because yeah, they are not so stable inside the cell, yeah? They're just used for protein synthesis and then they disappear. So are you targeting an RNA molecule that's actually disappearing from the cell? Is that a good strategy, you think?
Not really, but I think there's a moment if I read correctly, when the viral RNA is entering the cell, we can just intercept it in case we can use this metabolite just to prevent the RNA to enter the cell system.
Okay. Thank you very much for your defense and for your answers. Let's continue with the second presenter. So the second presenter, Eda Nur, is also including molecular docking studies and drug repurposing studies. So let's start the video then.
Hello everyone. Today I'd like to talk to you about integrative approach for melanoma. As we know, melanoma is a type of skin cancer and it is the most common of skin cancers. Since it is a difficult disease to treat, it is important to develop drug discovery methods. In this study, we applied drug repositioning for melanoma using a gene expression guided search tool, Genix Pharma, which provides gene-drug interactions. After determining the significant gene-drug pairs, we categorized them into 3 subgroups based on their relevance to cancer. The first of these is antineoplastics, which are used in cancer treatment. The second is drugs that are not intended for cancer treatment but are used as complementary drugs for cancer treatment in the literature. The third is drugs that are not yet used in cancer treatment. Our aim here is to determine the drugs that have not been used for cancer before, which can be used for melanoma. Here you can see what melanoma looks like. And so now we have come to material and methods. Firstly, we obtained the datasets for melanoma from geodatasets. Then we identified genes that differed significantly in the level of mRNA expression associated with the disease. Secondly, we identified the interactions of significant drug gene pairs in melanoma, and we did this based on the p-value. Finally, we grouped drugs in predicted drug gene pairs according to their relevance to cancer. And then we conducted molecular docking studies with the third group drugs that we determined. And here you can see a table showing treatments where drugs are effective for 3 separate groups. This is group 1, group 2, and group 3. And the disease they treat, as you can see. And here you can see 5 novel candidate drugs selected after drug repositioning and molecular docking studies. And as you can see, we chose venlafaxine, tenofovir, olanzapine, haloperidol, and doxepin. Normally, all of these drugs are drugs used except for cancer treatment. In detailed analysis, here we showed that the drug's binding site is compatible with the ligand binding site. We did this with 2 separate gene regions. This one's for 6-MOB and this one for 2HI4. In the 6-MOB PDB-coded structure, we see that the ligand and the reference ligand are compatible with each other. Here too, we see that the doxepin drug is docking to the right place as reference ligand. As a result, in this study, we propose new drugs for melanoma using computational drug repositioning and molecular docking studies. After all, we are planning to conduct cell line experiments for these 5 drugs, and we will do mRNA sequencing experiments for the 3 drugs that were successful in inhibition. That brings me to the end of my presentation. Thank you for listening. If you have, I can answer any of your questions. Thank you so much.
Thank you very much, Edanur, for your poster presentation. Professor Yelekci, do you have any questions that you want to ask Edanur? Eda, are you here, by the way?
Yes, I have one.
I'm here.
Thank you very much for your presentation. I have a little question. Did you do any work on inhibiting both enzymes at the same time, or are you planning to selectively inhibit one of them? Is there any drug among those repurposed drugs that inhibit both enzymes strongly at the same time?
Actually, we didn't do cell line experiments yet. So we didn't see any inhibition right now.
Okay. I mean, I'm asking computationally. You know, did you dock the same drug in different targets?
Yes.
My suggestion is that if you find something that inhibits both enzymes, that would be more effective.
I'm not sure. I can understand right now. Could you please tell me again?
Sorry?
Could you please repeat the question?
If you find one drug that inhibits both enzymes at the same times effectively, it would be more efficient. That's my recommendation.
Yes, you're right, I think. But we did just like that.
Okay, one by one, I guess. Okay.
Yes,
okay.
One by one.
Maybe you need to cross-dock and see the results, okay?
Okay. Thank you.
That's all. Thank you.
Thank you, Professor Yelekci. Dr. Serçinoğlu, do you have any questions?
Edanur, thank you. Yes, I have 2 questions and a comment, I guess. So Edanur, you have selected some possible targets for the molecules you want to test. And you do that with some tool called GeneX Pharma.
Yes.
What is the basis of this tool? How does it work? How does it find the protein associated with melanoma?
This tool based a pharmaceutical search tool. This is how can I explain? Sorry. This is the tool for drug and gene pairs, and we use this for gene data sets, actually, like geodata sets. It's similar, but its contents just 46 assays, includes melanoma, and we used this tool for the data sets.
Okay, so I have one small comment, but perhaps you may want to consider this for a future study. You know, when you take a look at the transcriptome profile in which a disease condition and a healthy person is compared, the proteins whose expression profiles change significantly are not necessarily drug candidates. Because, you know, a drug molecule may bind to some protein whose expression is not changed in a disease condition. So there is not one-to-one correlation. So perhaps the targets you have identified are not necessarily very relevant for melanoma. So then this might be some you know, source of concern for this. But this was just a comment. My second question was, so you have showed us a reference ligand for a protein and one of the drugs you tested against this protein, if I remember correctly, the binding free energy of your drug was lower than the reference ligand. So what do you think about that? Because
this is normal, actually. Yeah. This is normal because we want to be like that. We take the photo from PDB. I'm sorry. We take the gene structure from PDB, and it was with a reference ligand. And after that, we separate the reference ligand with a PDB structure. For example, 6-MOB. After we separated, then we dock with them again. You understand?
Yes, redocking. Yes.
Yes, we dock with them again. We dock with reference ligand and PDB coded structure like 6-MOB. We just want to see how it's I'm sorry. How it's
whether you can reproduce the docking post. That's basically what you want to say, right?
Sorry?
Whether you can reproduce your docking post. That's what you did basically.
Yes, reproduce. Actually, I mean that, yes.
Okay, thank you very much, Edanur. Unfortunately, we are over time. Dr. Sercinioğlu, thank you very much. Sorry for interrupting.
No problem.
But we have to continue.
Okay, thank you so much.
Thank you, Edanur. So our third participant is Atitya. It's a poster presentation, and I see it on the screen. Well, good luck, and let's start the video.
Good day everyone, I am Aditya, research student from Shastra University, India. I am here to brief our work titled Comparing the Effect of Glycerol and DMSO on the Dynamics of EcoR1 Enzyme, a Molecular Dynamics Simulation Study. Briefly, protein dynamics play an important role in determining its functionality. Knowledge of the structure and dynamics of the protein is essential for understanding their working in biological macromolecular system. To gain a complete understanding of the protein dynamics, it is important to consider the protein structure under physiologically relevant crowded environment. One such molecular crowder is osmolytes, the naturally occurring organic solutes within the living cells. Here we used type 2 restriction endonucleases, EcoR1, as a model system to address the effect of osmolytes in facilitating its binding specificity with DNA. EcoR1 has been used extensively in the molecular biology application owing to its high degree of specificity towards its cognate sequences. However, under certain non-optimal conditions such as presence of osmolytes like glycerol, EcoR1 has been shown to exhibit relaxed specificity called star activity, which is usually attributed to the osmolyte-induced dehydration of the protein-DNA interface. In this study, we hypothesized that in addition to the dehydration, osmolyte will affect the dynamics of the protein, which further together will bring about the changes in the specificity. For this, we investigated the effect of EcoR1 apoprotein dynamics in the presence of two osmolytes, glycerol and DMSO, at 5% and 10% volume by volume concentration. We performed molecular dynamics simulation with initial coordinates available from PDB ID 1ERI for 300 nanosecond using GROMACS. The dynamics of the protein was investigated using principal component analysis and the dynamic cross-correlation map. RMST, which is an indicator of stability of the protein structure, showed that the structure is maintained stable throughout the simulations, and the deviation of the upper protein structure is between 0.25 and 0.33 nanometer. Radius of gyration of the protein structure is between 2.52 nanometer and 2.54 nanometer, showing a compactness in their structure. Both RMSD and our radius of gyration showed that all the systems are stable throughout the simulation. Also, we calculated the average distance between regions of apoprotein units that are important in DNA binding to the protein, which includes enfolding arms, which is the interface of the DNA binding region, catalytic region, and the crosstalk regions between both subunits. Comparing the results with the cognate complexes showed minor variation in the average distances between the enfolding arms of the protein, which is 0.3 to 0.4 nanometer lower than that of the apoprotein structure. However, in case of catalytic regions and the crosstalk region, no significant variation in the average structure is observed. Residue-wise fluctuation showed higher values in the enfolding arms of 5% osmolyte system, whereas the fluctuation is lower in 10% osmolyte system, irrespective of the type of osmolytes. In addition to the residue-wise fluctuation analysis, dynamic correlation between all residues within the molecule called dynamic cross-correlation matrix was generated. The heat map from the cross-correlation matrix showed overall positive correlation within the subunits, including regions such as catalytic sites and the enfolding arms of the protein DNA interface respectively. Essential dynamics analysis was performed to capture the essential motion of the protein structure from its local fluctuations. Essential motion of the protein structure for the first principal component were visualized using porcupine plot, and the direction of the motion among the glycerol and DMSO at various concentrations is compared with no osmolyte protein structure. Based on the type of osmolyte, direction of essential motion for the both glycerol and DMSO system does not depend on the direction of the protein structure in the absence of osmolyte. In case of osmolyte concentration, directionality of the essential motion of the protein structure did not show any dependency when compared with the no osmolyte system. This infers that the collective motion of the protein structures are independent of both osmolyte and its concentration. The dot product of the first and principal component of the apoprotein structure, also called inner product matrix, with different osmolytes in different concentrations, showed root mean squared inner product similarity in the range of 0.4 to 0.5. Inner product matrix of the different osmolytes with respect to no osmolyte apoprotein structure showed variation in the dynamics of the protein structure in the presence of osmolyte. This confirms that there occurs a change in dynamics in the protein structure in the presence of osmolyte. In conclusion, higher fluctuation in the enfolding arms and the catalytic regions of the enzyme in the apoprotein structure with respect to cognate complex structure is observed. Also, we observed changes in the dynamics of the protein, especially in the enfolding arms of the DNA binding region, for both essential dynamics and DCCM analysis in the presence of osmolyte. We conclude that the osmolyte does induce changes in the apoprotein dynamics and may play an important role in facilitating the specific binding of protein and DNA. Thank you.
Thank you, Aditya, for this presentation. Professor Yelekçe?
Yes, thank you. It was a very interesting talk. I have one question. When you increase the percentage of DMSO, you are getting more stable dynamics, okay, instead of glycerol. What can you advocate at the molecular level that makes that kind of stability?
We were speculating from our other results that there is an interaction between water and the protein when DMSO is included in the interface than DMSO and the protein. Maybe we have to further analyze and quantify to confirm what we have identified so far.
My curiosity is that, is there any correlation with the dielectric constant of the solvent with respect to stability? We have any
we did not check
between the stability and the dielectric constant?
We did not check in that perspective, sir, but surely we will do that.
Okay.
Okay, sorry. Thank you very much, Professor Yedekçi. Dr. Sertinoğlu.
Yes, thank you for this nice presentation. So you showed us some essential dynamics results to us from principal component analysis. And there were some porcupine plots and I think some heat maps, right? So what do those heat maps actually show us?
Heat map is the pairwise residual fluctuation between subunits of between each subunits throughout the simulation, and we observed that fluctuation in chain A is highly correlated than the chain B. And this results also validated what we have observed from our RMSF broader calculation also. And we were to say fluctuation is mostly in the enfolding regions in all the cases of dynamical analysis.
Okay, so I think you can do one more thing. I assume you already have your data available. So perhaps you could also check the contribution of each mode of motion to the total variation from the principal component analysis. There's something like this. No?
Yes, sir. We are into that to understand which residue contributes more in overall change in dynamics of proteins.
So what I would suggest, take the modes that contribute at least 80% of the total variation in motion to get a more clear picture. Because from your principal component analysis, you showed us a comparison of individual modes, which were the first modes of motion, which indicates that this is the mode that contributes highest to the variation. This is good, but then in the next slide, in your heat map, you showed that there are like mode shifts. So in one condition, there is certain mode of motion. In another condition, there is the same mode of motion, but it is not the first mode of motion. This can happen with MD simulation. In order to overcome, the confusion, any confusion arising from this, there's a way to, assess multiple modes of motion by summing up their variances to summing up their contributions to the total variance. You can actually assess the overall dynamics between two systems based on a single metric from your root mean square inner product calculations. Perhaps you may want to check that, the methodology on this, how it is done. This was just a comment. Thank you for the presentation. Okay.
Okay, thank you both very much. So let's continue to the, this is the fourth presentation. Will be a poster presentation again from Jia Lim on ligand-based and virtual screenings on natural products against PDE3 for dual inhibitors. So let's start the video.
A very good afternoon to everyone. My name is Lim Jia, a graduate of Bachelor of Pharmacy from Malaysia. The title of my poster is Ligand-Based Pharmacophile Modeling and Virtual Screening Studies for the Discovery of Novel PDE3 and 4 Dual Inhibitors for COPD. To give a bit of a background, COPD, or Chronic Obstructive Pulmonary Disease, is a progressive lung disease primarily caused by smoking. It has seen a steady increase in prevalence over the recent years, and current treatments of COPD, such as beta blockers, cholinergic antagonists, and inhaled corticosteroids, are aimed at symptomatic relief and provides no means of a cure. On top of that, prolonged corticosteroid use also makes the patient susceptible to infections and other side effects. Recent studies have hinted at the potential of PDE3 and 4 dual inhibitors as a new approach to COPD drug treatment, which may produce a higher efficacy and better safety profiles. In our study, we have employed computer-aided drug design techniques to achieve the following objectives. Number 1, to develop a ligand-based pharmacophore model for PDE3 and 4 dual inhibitors. Number 2, to perform virtual screening of the UNPD database to identify new PDE3 and 4 dual inhibitors. And number 3, to carry out molecular docking studies to investigate the ligand-receptor interactions at the active site. The methodology of our study can be split into 5 main stages, namely pharmacophore modeling, virtual screening, molecular docking, visualization, and structural modification. To develop a pharmacophore, we gathered known active ligands from literature, clustered them based on their structural similarities, and generated pharmacophore model using Ligand Scout Advanced. The pharmacophore model was then validated and screened against the Universal Natural Products Database or UNPD database, followed by a drug likeness analysis. The best hit compounds were docked into PDE3 and 4 protein structures to observe the interactions that occur at the binding site. And in the last stage, we proposed modifications to the best hit compounds to maximize binding affinity, optimize their physicochemical properties, and also to increase the synthetic feasibilities. The results of our pharmacophore modeling yielded a highly specific and sensitive pharmacophore model, and Figure 3 shows an active compound superimposed onto the validated pharmacophore model. As you can see, the pharmacophore model has 4 hydrogen bond acceptors shown in red and 1 hydrogen bond donor shown in green. Virtual screening returned a total of 345 hits that matched the pharmacophore, and through drug likeness analysis, we have identified 20 filtered hits. In the next stage of molecular docking, each of the filtered hit compounds were docked into PDE3 and PDE4 proteins, and the compound UNPD1558 shows the best overall binding affinities for both proteins. The figures to the right illustrates the structures of the proteins, as well as UNPD1558 binded to the active site of each protein, PDE3 and PDE4. Based on the SAR of UNPD1558, we were able to propose several modifications to its chemical structures. Out of over 50 derivatives that we have come up with, compound 25A4 showed significantly improved binding affinities towards both PDE3 and PDE4 compared to known active compounds and reference compounds. In conclusion, our study identified 2 lead compounds, UNPD1558 and compound 25A4, that showed superior binding affinities towards the PDE3 and 4 proteins. This highlights their potential as the new drug approach to COPD treatment, and further studies in vivo and in vitro should be carried out to confirm their biological activities. Thank you.
Okay, thank you very much. Very interesting topic. Let me start this time with some questions, very short questions. I like your presentation very much, but there are some questions that I would like to ask. If you look at the middle column at the lower part where you discuss and show the results, you say that you have validated your pharmacophore model, but you don't show any numbers here. So could you share the numbers with me? I mean, how many hits and active, decoys and actives have you screened with the pharmacophore, and what were your retrieving numbers, your enrichment?
Yes, all right. Thank you very much for the question, Dr. Attila. So regarding that information, because we didn't have enough space on the poster, so that I didn't include, but I have it here. So from our selection of active ligands, we selected 28 active compounds from the literature. 21 of them were included into the training set. And then if I can Give me 1 second to bring out.
No. Just we're just talking about your poster.
Right.
So give me the numbers already.
21 was in the training set and 7 of them was in the test set. So after validating the best pharmacophile model that we have, the parameters was sensitivity of 0.857, specificity of 1.00, and GH score, which we used to assess the pharmacophore, was 0.964.
Okay, so then you do some docking studies and you selected some possible hits, right?
Yes, correct.
So have you also validated your docking procedure?
So for the docking procedures, we have selected the top 20 hits from the screening, and then we dock them based on the, well, first we have to select the protein structures from the PDE database. So we selected them based on their specificity to PDE3 and PDE4, and then as well as having a co-crystallized ligand with a very low IC50. So a very low IC50 will allow us to basically mimic a tight binding site conformation. And based on that tight conformation, we will use it as a basis for our docking studies.
Yes, I see, but did you perform some enrichment graphs to see again with hits and decoys? I mean, actives and decoys?
For docking, no, we have not.
Okay.
Yeah.
In these kinds of virtual screenings, it's important that you can rely on the post that you create with your docking procedure, and also that you can rely on the score, because you're doing the prioritization of your compound library based on the score also. So if you can't rely on your score, then it is this procedure is very risky. So therefore, I advise you to perform a very sound validation of your docking protocol, so you can rely, or at least on what percentage of the, how strong you can rely on your scores. That's basically what I wanted to say. Thank you very much. Professor Yelekçi, do you have any questions?
First of all, thank you very much. And I have a very quick question. PDE5 has a very similar structure to PDE4, okay? They are coming from the same family. Have you ever tried some of your best compounds against this enzyme, PDE5, because it has also a very important biological activity, and see the selectivity?
Thank you for the question, Prof. No, so far we have not, because most of our hypothesis was from the previous studies, which showed there's actually previous PDE3 and 4 compounds that have been tested for COPD, but for PDE5 we have not tried it. We can consider trying it in the future.
Okay, if you get some efficient inhibition, you will be surprised with the results, okay?
Okay, all right. Thank you, Prof.
Thank you very much, Professor Yelekçi. Dr. Serçinoğlu, have you any questions?
Thanks for this nice poster presentation. I don't have any question, but I would just like to say I agree with Professor Attila's comment. That you need some validation of the docking protocol here, because taking a look at your results, the difference between the reference and the modified hit compound is like 0.5 kilocalories per mole. We should always keep in mind that molecular docking does not produce very accurate binding free energies. It's a way to filter candidates from a large pool of molecules. So yeah, you may want to consider that before you move these molecules to in vitro tests. Thank you.
Okay, thank you very much for the advice. All right, thank you very much.
Okay, thank you. So now we have an oral presentation of Pamela Miranda. Okay, I see it on my screen, so let's start it.
Hello, my name is Pamela. Today we present you our work about PCR primes to detect SARS-CoV-2 variants of cancer. Polymerization reaction is a molecular technique used in several applications. One of its elements is primers, usually a pair of primers called forward and reverse or sense and unsense. Primers are short DNA sequences designed to recognize genomic regions. PCR has three main steps. The first one is the denaturation, in which double strands are transformed in single strands. The second one is the annealing, in which the primes, in red here, have latched to the target. And the last one is the extension step, in which the DNA polymerase amplifies the product. These three steps are carried out from 30 to 50 cycles, and each one occurs in a different temperature. In our work, we are focused in the second step, the annealing, when the primers have latched to the target. And it's the moment when mismatches may occur. So what are mismatches? It is known the canonical base pairs AT and CT. So all the different mismatch of the different base pairs are mismatches. That is an incorrect pairing when a base binds to a non-counterpart, and there are 8 different mismatches. Mismatches are usually associated to this destabilization of the molecular structure, changing the conformation of hydrogen bonds and stack interactions. As, for instance, in red is the GA mismatch base pair in comparison to GC base pair in gray. So we can see a slight difference in the conformation. And the presence of mismatches also influence in the melting temperature. However, some types of mismatches are more stable than others, even more stable than AT canonical pair. Because of our work about primers, it is known the presence of a few mismatches in primers may improve the stability of hybridization in comparison to AT-rich primers with no mismatches. One factor can affect the mismatch are the neighborhood. So the base pairs which flank the mismatch pair in both sides. There is 3 types. The first one of them is when the mismatch is flanked on both sides by canonical base pairs. The other one is a mismatch base pair and a canonical base pair. And the last one is when we have a triple mismatch, a mismatch base pair flanked on both sides by mismatch base pairs. So in the 2 first ones, we can see the same primer hybridized to the sequence. But in contrast to what we expected, the presence of mismatch decreased the melting temperature. Here we see a different situation. When we have double mismatch, the melting temperature increase in comparison to the same primer with a single mismatch. However, this data is not simple, and more calculations are needed to improve the understanding about mismatch and melting temperature. And some mutations may induce the presence of mismatch. For example, in our study about SARS-CoV-2, in regard to original genome, we have no mismatch in this example. But with mutations which induce the variants of concern presented here, mismatches come occur. So here we have some examples from our work with mismatches. So from this Our question is, are these PCR primers able to detect SARS-CoV-2 variants of concern and not fail the diagnostic tests? We collect 90 prime probe sets of PCR from Charité, WHO, and CDC, and genomes of variants of concern, alpha, beta, gamma, and delta. We align them primes against genomes using the Smith-Waterman algorithm and consider two strand configurations. One, we have the genome 6 like in the FASTA file, and the other one, the complementary genome sequence. We calculate the hybridization temperature of primes and probes, which call the reference melting temperature, and the meta-alignments, that is only canonical base pairs, and the mismatch alignments, considering up to 3 consecutive mismatches. For this, we use the recently available thermodynamic values for DNA mismatches. And we calculate the coverage for meta-alignments and the mismatch alignments. Here, considering a limit of temperature, which is when we have the difference between reference melt temperature and the temperature of the mismatch alignments. If its difference is below or equal the limit, we consider this alignment. This prime is acceptable. So, summarize our workflow, we have the alignment of primes and genomes, the calculation of hybridization temperature, and the calculation of coverages. So what we found a substantial number of primes achieve coverage above 90%. And when we consider the mismatch, most primes achieve at least 99%. Some, for example, CORMA from new coverages for original genomes achieved more than 90% considering mismatch. For example, this primer from WHO achieved the high coverage considering mismatch for all 4 variants of concern, alpha, beta, gamma, and delta. So beta, for example, from 98.1 to 98.6, and delta variant achieved 100% when mismatch were taken into account. This probe from Institute Pasteur, we see for variant alpha variant a 99.7% when we consider method alignments. However, when we look to delta variant, this coverage is slightly low. But when we look to mismatch coverage, delta variant achieve 100%. It is 84 more alignments, about 1% of all genomes of delta variant we collect. From both alpha and delta are about 7,000 genomes collected. So answering our question, we can say when we consider the mismatch, the coverage increase for variants of concern, so maybe it would be good to the detect of virus. In particular, when we look to delta variant, a few cases achieved 100% for mismatch alignments, with what it suggests these primes should continue to be effective to detect the SARS-CoV-2 variants of concern. Our software is available in our site, so feel free to use it. Thank you to Financial Support, Recaps, and CMPQ. And thank you for listening, and feel free to send me a message. Thank you.
Thank you very much, Pamela, for your presentation. Professor Yelekçi, do you have any questions for Pamela?
I thank you very much for this nice presentation. I have one more question. So is there any correlation between the mismatching percentage of these primers with the efficiency of DNA polymerase reaction?
Yeah.
So what is the minimum error that the DNA polymerase work efficiently?
Still, we can't get to do the clinical validation, but we work with graphs, which shows when the maturation starts. So some cases, the mismatch has a good melt temperature, but it's destabilized the free point. So we are investigating now if this contribute or it may disrupt the polymerase action. So still we cannot answer the correlation.
I see. So I just wonder if there is only one mismatch, maybe the DNA polymerase work effectively. When you increase the mismatch number, then the enzyme will probably have difficulties. But I don't know the lower limit of that.
Yeah.
Okay. That's all. Thank you.
Thank you.
Thank you, Professor Yelekçe. Dr. Serçinova, do you have any questions?
I don't have any questions. Thank you for this presentation. It was clear and nice. Thank you.
Dr. Serçinova, thank you very much. Pamela, thank you very much for the presentation. And let's now continue to our final attendant. Let's find the documents. Okay, so the final one is entitled Blood Group Susceptibility Shifts in COVID-19 Variant Infection Rates by Atreya. So I see that's ready, so the floor is yours.
So a good day to all the viewers tuning in to the 5th International Symposium on Bioinformatics. This poster is focusing on blood group susceptibility shifts in the COVID-19 variant infection rates. So we're basically taking a look at how different factors and protective mechanisms play a role in both the transmissibility and lethality of COVID infection. So initially, during the first COVID outbreak, when the entire crisis started, the mechanism was investigated, and it was found that the glycoproteins on the surface of the virus do bind to the blood cell surface receptors, and the specific component that does is the S1B part, which binds to the angiotensin converting enzyme 2 receptor, which is one of the most significant parts of the infection itself, as it leads to both entry into the lung epithelial cells as well as the overall hypertensive effects of the illness. So over time, as different studies were done regarding the relation between ABO blood groups and the COVID-19 severity, we were able to find that the viral infectivity has been linked to both differences in geographical and demographical factors, but a blood-dependent infection rate has been noted by several studies because the population differences when it came to the blood group frequencies were very evident. And the DNA genotyping has shown that A blood group individuals have had a higher severity and a higher transmissibility when it came to the COVID infection compared to other blood groups during the first wave, especially when compared to those of O blood group. And this tendency shifted from each wave and from each variant, as during the Delta virus and the resultant wave, we were able to find that individuals with B blood group were slightly more susceptible. And then AB blood group also showed increases at that point. And in general, the O blood group stayed more or less the most neutral when it came to susceptibility. And the reasons for this were explored and different mechanisms were pointed out. The study evidence showed that those with anti-A antibody production often showed a protective mechanism against COVID-19 infection, as the antibodies blocked the binding to the ACE2 enzyme receptors, thus blocking entry into the lung epithelial cells. And this also played a role in the microbiota because bacteria actually induce the synthesis of anti-A antibodies. Now you can ask, there are different blood groups with the same anti-A antibodies. However, the anti-A antibodies that are required to block both binding and overall infection are of the IgG type. So this blocks the binding to the S proteins and thus the glycoprotein infection itself, versus those of people who have B type blood group have IgM antibodies that are anti-A. So that's less effective during the infection itself, and thus those with B type blood group did show an increase in subsequent waves compared to those of O blood group who had IgG antibodies. And the understated theory is that through natural selection and high existing polymorphism in the ABO blood grouping plays a significant role in the infection itself, since the natural selection plays a role in different illnesses across the world and different outbreaks, including malaria, where blood group expression has been shown to increase prevalence in certain areas compared to others. And the subsequent waves have also shown differences in oxygen hemoglobin affinity. So that could have also been an effect that played a role when it came to those of different blood groups. But more than this, the rhesus factor negative individuals have shown a much lower incidence of COVID-19 than those of higher rhesus positive blood groups, as well as a higher incidence when it came to higher severity of infection. When you look at the statistics, those who had AB blood group showed a much higher susceptibility in the subsequent waves. And the difference in intubation rates and the lethality rates also changed. They were lower in the A blood group individuals at one point in 2020, and they increased in AB and B blood group individuals at that time, versus the death increased in AB blood group individuals and decreased in A and B blood group individuals, telling us that it does change over each variant in each wave. But the protective effect still has shown a large effect when it came to the O blood group individuals, possibly contributing to the severity of infection. And this is attributed to different factors, including lower surface area on the surface of O blood group individuals, and thus lower binding affinity toward the infection, as well as other kinds of receptors and anti-A antibody production. So the conclusion of the study is basically that the ABL blood grouping has shown a significant association and link to both the infection rates, the severity, and the intubation of COVID-19 infections. And this has changed over the different waves. And this is to be explored because the anti-A antibody production, if it can be replicated, it could give the protective effect to people all around the world as a possible buffer in the next wave or the next epidemic, if we can note the same kind of pattern. So it is definitely something to consider in significance. And I hope everyone who's been watching will stay safe in the future epidemics. Thank you.
Thank you very much for the presentation. So Atreya, are you here? Do you hear me? Okay, Miss Cholak. Is she here?
I think she's not.
Okay, so in that case, if she's not here, she was the final presenter of our parallel session today. So there's nothing we can do. I thank Professor Yelekci and Dr. Serçinoğlu for their attendance and for their high quality questions. I thank the participants for their presentations. And well, let's see each other after lunch break. I think I give the floor back to you.
Yes, thank you, Professor Dr. Attila, and our two honorable judges, Professor Dr. Kemal Yerekci and Professor Dr. Onur Selçinoğlu. Please do fill out the forms for these oral and poster presentations. I would like to thank all our authors who put their efforts on these oral and poster presentations, and all our participants for being here with us. Now CADD and SARS-CoV-2 presentations are over. Big data analysis and biotools and general bioinformatics presentations is going on now. If you want, you can change your breakout room to join there. After these presentations are over, we will have the lunch break and we will meet at 2 p.m., which is going to be Turkish Standard Time. The participants are invited to attend our next parallel session. Then we will have a keynote talk later in the day by Dr. Yuval Tan at 4 p.m., which is going to be Turkish Standard Time. See you there. Thank you all.
Okay. Thank you. See you.